Hello everyone. Today I'm gonna to present my work is entitled by Insect Infesting Stored Food Product, Confused Flower Beetles, Tribolium confusum dival. Before to start presentation, I'm going to present myself. I am Rekia Sharif from University of Gardaya in Department of Biology in Gardaya also University. I am Associate Professor Class B. As introduction, the Sahara region of Algeria represents one of the most remarkable reservoirs of flora and phytochemical diversity, hosting a variety of spontaneous plant species that have evolved unique chemical defense mechanisms under extreme environmental conditions. This part of this study highlights the insecticidal activity of Saharan plants against Tribolium beetles is a major pest of stored foodstuffs. The originality of this scientific research is evident of several levels. Firstly, the plant studies of endemic to the Gardaya region. Also, the process applied both of extraction of a bioactive compound and for experimental treatment. In this context, our review indicates the research of insecticidal activity of extract aqueous of Cleome arabica were harvested in Sahara Algerian. As methodology of our research through of the production secondary plants regulate and define themselves against their environment content. In the plant insect interaction, this compound may act as anti-appetite, toxin, and anti-fertilizing agent, or allelopathic as plant with other plants interaction. So our study is based in the evaluation of insecticidal activity effects of Cleome arabica. So let's describe our plant, which spontaneous plant which reaches for 50 centimeters with the purple flowers and longinated fruits. For the analysis test, the leaves of Cleome arabica were collected and properly rinsed with water to remove any material stick to the leaves. After that, they are separated and put it in the pack in the vacuum room. Also, when the leaves are completely dried, they are crushed by using electric grinder to obtain the thin homogeneous powder. The powder leaves is used for reflux extraction with 100 grams powder in solvent mixture composed with methanol and distillate water. After cooling this solvent, the filtrate separate from the solvent using the rotary evapor. The final extract is aqueous extract used for the tests. For the test, we use 10 concentration, and also the negative control is distillate water. The positive control is insecticidal product. For the ingest protocol, it consists for 5 of semolina to 10 adults of confusum dival beetles placed individually in sterile petri dish. The insects were treated with 4 ml of aqueous extract for the positive control with 1 ml, and for the negative control we use 4 ml. As the result, we calculate mortality rate, lethal dose, and mortality kinetics. For the first parameters, mortality rate for higher concentration, the mortality rate is more than 80%. For the lower concentration, the mortality is so moderate. For the little dose, the result group showed the value of the little dose, which are defined by higher ingestion and contact toxicity of this aqueous extract of Cleome arabica against Tribolium beetles, which are 0015 for DL50 and DL19 is 0051. For the cynetic mortality, the mortality is observed after the 3 days for the higher concentration, which the mortality rate is more than 10%. For the lowest concentration, the mortality is observed just for 60 days. So, as conclusion for our study, this result is a very important study which demonstrates that aqueous extract of Cleome arabica exhibits a significant insecticidal activity against Tribolium confusum. Of also, this finding supports further investigation into development and plant-based insecticides as environment-friendly and sustainable pest control agent. As acknowledgement, I would like to thank all the organizers of this conference. Also, they also extend my gratitude to audience for their attention, interest for the thoughtful question, which grateful contribution. I'm thankful for my team in university laboratory as HEROUINI Amel, doctor in my university. Also, Monsieur KEMASSI Abdellah for this university. Don't hesitate to contact me.